Anat Ben-Zvi. She is the chair of the Life Sciences Department. She's the chair of my department. And she will talk about the exceptions to the central dogma of biology, bending the rules and extending the story.
Okay. Thank you very much for staying, and I hope this will be interesting to you. We're discussing with Ute about what would be interesting to this crowd to discuss. We were trying to think, I was trying to think about how do we really view science and how do we think about science in an everyday life, and how is science progressing from one discovery to the other, and how is it shaped, and basically asking ourselves whether or not we can really talk about revolutionary revolutions in science. And so, to sort of challenge my thinking about it, I went to something that had come up every time I teach Cell Biology 101, and that's the central dogma in biology. From its name, it's pretty clear it's very essential. And also one of the things that you probably noticed, if you found it, it was mentioned many times in the various biology talk as like a central theme that we're working around. But to understand the central dogma, you really need to understand the context, and so I'm going to take a few years back, not much, and talk about the discovery of the DNA. And the most important aspect of the discovery of the DNA, apart from or let's say there are so many different aspects of why it's so important. One of them is the birth of molecular biology, but the other thing is the intuitive nature of our understanding of the chemistry of DNA in the sense of how it can store information. Because once you look at the DNA structure, you realize that the basis in the DNA, the part that is different, the letters of the language that has been described here, the 4 bases, can only pair in one particular manner. And so once you know one letter, you know the other letter on the other side. And this is important because once you open the DNA and basically allow, let's see if I can, this is fine, yes. Okay, so once you open the DNA and you have now a template on which you can build a replica that is exactly well, basically you create 2 replicas that are exactly the same information that was in the original strain. And that basically means the DNA, once you understand its structure, fulfill the main function that is required for self-replicating material, and that is the ability to create another copy that is exactly the same as itself. And so this was crucial, and once it was sort of suggested, and that basically part of the original paper was that if you see the structure, obviously this is correct, and then not very far after that it was shown experimentally that indeed if you synthesize DNA using light isotope and a heavy isotope and then allow the 2 to mix, you get an average, which means you basically created a sequence that is one strand like this and one strand like this. And so this has really revolutionized the way we are looking at molecular biology, and basically introduced something completely new, sort of solidifying the idea that DNA is the genetic material that we sort of knew that was functioning there. But then what we, or let's say what science was trying to think is, okay, DNA is there, DNA is the information, but then how is that information turns into what we actually see as a biology function as proteins. And proteins were known, enzymes, and we know this, but then how do you take DNA and then transfer the information into proteins? And one of the main challenges here is the DNA is localized in eukaryotic cells in the nucleus, so it has its own compartment, okay? But translation, or the production of protein, is mainly occurring in the cytoplasm. So we have 2 different areas in which those things happen, and we need to sort of bridge this in order to understand how these 2 worlds are talking to each other. Another challenge here is those are 2 very different chemistries. So DNA has bases, it has 4 bases. Amino acids, which is the basic units of the proteins, have around 20 different ones. They have very different chemistry, and so how those two chemistries are talking to each other has been one of the major things that people were discussing. And there were all kinds of crazy ideas. So the DNA create a template on which you all kinds of ideas that trying to think about how those two molecules can talk to each other. And here comes Crick, the pair of the Watson-Crick, the guy himself, trying to think about how this is happening, and he presented this concept in a talk he gave in a lecture, 57, titled Protein Synthesis, but it's everything but just not protein synthesis, talking about one of the basic ideas that we now view in biology, which is the flow of information in biology. And he suggested that DNA have the information. That information can be made to a copy in a form of RNA, which is more transient and less stable, which can then be translated into a protein. Okay? He says, and I put this quote because I really liked it, that everything he says here is completely hand-waving, okay? Because the data, there was no messenger RNA observed, no tRNA and soft ribosome. It wasn't really clear what they're doing. There were a lot of pieces missing here, but he thought that if you need to translate this information, if you need to create this gap, this is the way it's going to happen. And he says that I will argue that the main function of a genetic material is to control the synthesis of protein, basically to code for those proteins, as we call it today, and that this, without evidence to support this, he thinks that this is what should drive the hypothesis, and should be basically, even if you don't have an independent. And because he thought it was such a breakthrough hypothesis, he called it the central dogma. And I will meet in a second the idea of central dogma, but I want to make clear one of the things that he said and has become like sort of like main thing in the central dogma, and that he suggested that the flow of information can go DNA, RNA, protein, but it cannot go back. And the main idea is that once you translated the chemistry of bases to amino acid, you cannot translate back amino acid into a basis, and so you cannot go in reverse. And so this hypothesis basically postulated first the idea that there is transient RNA molecules that can be copied based on the DNA and transfer the information before the discovery of messenger RNA, which is basically taking advantage of the fact that we can open the DNA, create another molecule which is shorter, and make this as a copy that they can then use. And the most important concept here, which is the adapter, or basically how we translate one language to the other. We use a molecule called tRNA, that on the one hand has what we call a codon, or a read that can read the code of the sequence, and on the other side has an amino acid attached to it. And he already suggested, again, without any information, that the code should be 2 or 3 letters. Makes sense if you want to code 20 amino acid bases on 4 bases. And then that there is some kind of an enzyme that puts the correct amino acid on this, and so create some kind of a translator. If you want to think about it, it's like if we have an adapter for electricity in Britain, and you want to put your European one, and so you need something that will translate one to the other. And so this is basically an adapter between the two languages. One side of that molecule read or produce or interact with the DNA RNA, and the other one is basically the protein. And so then when you think about protein translation in the end, what you have is you have a message molecule that carries the information. That molecules have the bases. You read those bases in Windows. We read them in codes of free. We have those translator adapters units, the tRNA that reads on one side and introduce the protein and the amino acid on the other. We create the peptide bonds and we build a protein. So basically, this is the concept of DNA, RNA, protein. And here is where biologists make a lot of mess, okay? A lot of students say DNA, RNA, protein as if we are transferring, this I don't know, you make a sandwich, I don't know, you mix and match. It's the information that is flowing here. And here is also the idea of that I myself did not know about central dogma, because I thought that what Crick actually suggested that is that information only flow in one way, DNA, RNA, protein. Okay? Turns out that this particular image that is in every book in cell biology you look, okay, was drawn by Watson, and it's his interpretation of the central dogma. Okay? Which, again, what Crick says was that if you have a protein and you created the transfer from the chemistry, you cannot go back. Okay? He actually, if we go back for a second, did not say that you cannot do DNA to DNA, DNA to RNA, RNA to DNA, only that once you have a protein, you can't go back. We can still argue about that. Yeah. Okay, and another thing that I have to sort of make clear, again, it was a surprise for me, apparently. Some people would think about it. I've known that, but again, as a teacher and a biologist, I did not know that. That Crick was criticized about calling it.
Can you speak a little closer to the
Yeah, sorry. So he was criticized for calling it a dogma, okay? And then it turns out that by saying, and someone said earlier commented about shared language, okay? So when he said dogma, he meant a bold hypothesis with no proof. Okay, whereas most of us, when we say dogma, we actually mean something that does not require anything but your belief in order to have, and you basically cannot change. So if we want, we can call it a profound hypothesis. I actually think, making this talk and thinking about this subject, that it's sort of like an organizing principle for modern molecular biology, if we want to suggest one. Okay, so now, and a few other speakers that sort of relate to this. Does the dogma stand? Okay, is it a principle that is standing? And what do we do when we challenge this hypothesis? And when this hypothesis seems to be invalid? And do we now reject this and take another hypothesis and work with a different one, or how are we responding to changes? And I'm going to give 3 examples. There are so many others, but I want to focus on 3 different examples. Some of them we sort of heard and related to, but want to a little bit dig into. So the first one we heard in a previous talk referring to viruses and RNA. So one of the earliest discoveries was that, looking at this context, is that you don't necessarily need DNA as the template of information. You can use RNA. And then not only that, that you can take RNA and turn it into DNA. And that's the concept of reverse transcription, where you go from RNA to DNA. And immediately when it was discovered, someone who's remained anonymous wrote central dogma reverse, because if we can go back, we can go back. Crick made a point of saying, I did not say that you cannot do RNA with DNA, I just said that the protein, if you talk in a language of protein and you go residue by residue, you cannot go back from a protein to a nucleic acid. Okay? And it is true, at least in a sense, that you can code one amino acid in more than one sequence, and so you can't really know exactly what the sequence was just by knowing the amino acid sequence. You can make variation on that. But again, so this idea, and I think other speakers have also noted that one of the things that this made is a major shift in the way we do science and biology, because it introduced the ability to create cDNA, basically take RNA, make it a DNA, and then do a lot of other modification or a lot of other work on it. One of the major things is cloning and creating our own engineered DNA molecules, which then gives you a lot of tools that you can ask positive questions in biology. So really turning around biology in a term that we don't just observe nature, we actually change it and then follow what's going on. The second example, again, was referred to by the previous speaker, talking about splicing and RNA editing. And this is, again, we're saying DNA, RNA, protein, we're talking about linear information transfer, but then here we have an option of actually cutting some of the information out on the way to the RNA. And so where is the information about which part is being cut? And this information is not just saying make a protein, it's actually saying make the protein this way, cut these sequences, and this information, as was pointed by Ramon, could actually change between tissues, can be different in various circumstances, times, and regulation. So it's a very intricate control unit that can change the information and the way it's actually put into practice. This again goes into the eukaryotic genome. I will not go into too much because it was discussed, but you can do splicing, you can do alternative splicing, you can put modification on the RNA, you can change its lifespan, you can change a lot of different factors that can then impact gene expression. And so, even more further than that, you can take the RNA that you created and introduce a protein that will edit that RNA. So introduce new information that was not in the DNA at the RNA level, RNA editing. So you have a protein, comes to the DNA, change the base in the DNA, and now you have a different protein. And that different protein can be either because the RNA now produces a different frame shift, and so we read the protein differently and create completely different protein, or we change the splicing, or we change any other option, or we just change one amino acid. But in the end, we change the information. So we can change the message, and so change the information, but again, we did not change the flow of information. The flow of information is still DNA RNA, although we can introduce information in other ways. And finally, I want to talk about prion biology, which is sort of closest to my research, which is protein folding and protein misfolding diseases. And I think that is one of the most interesting concepts in this way, because this has challenged the field in several ways in terms of how do we view information flow and what is exactly information. And so to introduce the issue, I need to introduce the disease that sort of led to the discovery, and there are two main ones that you probably have met. Mad cow disease, to those of us who were here in the Russia in the 90s, that was a very interesting idea and concept between you and your hamburger. And basically the idea that you can transmit disease through processed meat. And one of the interesting aspects about it is because it did not fit to any form of infection diseases that we knew. So it was infectious. You can get an infected material and you can transfer the material or the disease to a new organism, but it wasn't a bacteria and it wasn't a virus. And if you try to kill it in any ways that kill DNA, it wouldn't care less. But if you did, or you use techniques that interfere with protein folding, protein stability, mostly severe one, but still, you can actually reduce the infectivity. And so...
Sorry, can you repeat that? I didn't quite get which techniques do.
So if you try to use technique or agents that kill protein, treatment that kill protein, not everything, because those proteins are actually pretty resist, so you can try degradation and it doesn't always work, but some techniques that are basically causing unfolding of proteins, those will cause you to lose the infectivity. Okay? And so Krusner suggested the concept of prion, proteinaceous infectious particle, and suggested that protein can be the infectious agent. Now, the idea of a protein being an infectious agent is, on one hand, interesting. On the other hand, it really says, okay, if you are an infection agent, what you're actually doing is transferring information from one organism to another. And so that means that the protein can transfer information. But then how does it do it without the DNA? And so there were some scientists that spent quite a lot of time trying to find the DNA in those proteins, claiming that like a virus, you have a capsid and the DNA or the RNA molecule is there and it's just small and it's unable to detect it, and so we have to go and find it. Okay? And it was a huge debate. I think I told Ute once, my mentor, my PhD mentor, went to a meeting and someone in the crowd says that when will you finally accept that there is a DNA there, and the guy at the podium answered, when people like you will die. Which I think is a misunderstanding of Kuhn, if it ever was one, but also I don't really, it really created a tension in a sense that this does not relate to anything that we understand in terms of DNA, RNA, protein concept of information transfer. And so the main question was what kind of information is transferring here and what is exactly going on. And for this I need to explain one thing about protein biology that I sort of ignored and glimpsed through when I was giving this talk. And it is that one of the major questions of how you translate information from DNA to protein is a spatial problem, because DNA is linear, RNA can be, but mostly the information is linear, whereas protein is three-dimensional moiety. And so for the longest time, many of the hypotheses of how you can translate information try to deal with the conformational organization of the protein. Crick solved that problem by simply making it very simple. So he said, let's just assume that the information that is required to get the three-dimensional structure is already embedded in the first sequence, the linear sequence, and if that is correct, then all I need to do is transfer the linear information from one chemistry to another chemistry, and from that point on we will get the three-dimensional conformation. And for the most part, this is Anfinsen experiment, another one of those basic principle in biology, and that's a basic proof of this concept. And what he did is he took a protein, denatured it, so made it unfolded completely, then diluted the agent was causing this unfolding, allow the protein to try to see if it can reach back to its three-dimensional structure, and in sense got it. There are not some proteins that can do that, but there are proteins where many proteins can, and sort of phrase this concept of all the information that is required to define a native structure is encoded in the primary sequence itself. And so then what kind of information is carried in a protein if a protein has this conformation and the protein is now infectious versus not infectious? And one of the interesting concepts in this field was the revelation that there are in fact two structures from the same sequence. The first structure is a folded structure, normal structure, which is actually carrying the function of that protein. And that is a normal protein that exists in many different organisms. And the second structure is a different structure using different structural moiety, and that structure creates big oligomers of proteins, we call them protein aggregates, huge structures that interfere with protein, with cellular function. Okay, so basically what we have is we can have a shift between two structures, and one structure can carry information to another one. And so the idea here is that the information is not in the primary sequence and is not interfering between the information that came to the DNA, but it's the interpretation of that information in the three-dimensional structure. You can again argue whether or not it's interfering with the logical flow of information, where is the information coming from. One of the interesting aspects of this is that the protein aggregation concept is the basis of many different diseases. Prion biology is an extreme where you can actually infect the protein, and there are some examples like Kuru, where you have people that in Papua New Guinea that used to resort to cannibalism. They would eat the brain of their dead as an honor, and that will pass the disease. But also Jakob Kreutzer, which is a genetic disease that can be transferred, for example, from a patient to a doctor by a cut. Okay, so you can actually transfer, again, the agent and then cause the disease. Other diseases are not known to be infectious, but you can see the infectious behavior in a sense that one conformation of a protein can actually produce other conformation of the proteins, and that is true in Alzheimer's disease, Parkinson's disease, and ALS, and so on, which all share this basic structure of filaments or aggregates that are interfering with cellular function, but they are caused by different proteins, they are caused in different tissues, and cause very different diseases. One of the interesting aspects of those filaments is the fact that they can create different structure, and so basically the same protein can create aggregates, but not just one aggregate, but multiple aggregates. You can create many different types of aggregates, so there are a lot of information that can be stored here, and the information can produce many different phenotypes. So it could produce different disease, it could affect different areas of the brain, it could affect interaction with other protein, and so it could change completely the way the disease is moving, even though the protein that was coded in the DNA is the same. I saw a lecture recently by Ronald Mulkey that made this interpretation of looking at those filaments as like pasta. Okay, pasta is all made out of the same thing, but you can have many variations of pasta. They don't taste the same, though it's the same. It's the same, but the main reason they differ is because their interface with the environment is different, and so the amount of sauce on the pasta will be very different, and so the interaction with the environment will be very different, and so the impact it will have on you tasting that pasta is going to be different. So in a way, you can think about those feelings as the same. So basically, if we go back to the idea, this was a lot of created a lot of tension in the field. But in the end, it was just resolved in a sort of a sense that, okay, this is part of biology, this is a variability, and so this is one of the things that we're now incorporating into the concept of how you can transfer information in biology. I wanted to introduce one more concept, which when I was thinking last night, I was like, oh wait, what about CRISPR? Because what is CRISPR? CRISPR is a protein that can go and edit DNA, and so here we have a protein that can actually change DNA and therefore can change the flow of information. But then if you think about it, CRISPR doesn't work without nucleic acid. It requires a guide to go to a particular place, it requires a template in order to actually introduce new information into the DNA. And so it's an enzyme, but in the end, we're still in the world of DNA. And so this brings us back to sort of the challenging of the central dogma. And if I think about it, the way I view science, and it's that we have organizing principles that help us think about biology. They don't necessarily describe all the situation, because one of the amazing things about biology is that the only rule that you can, even that rule doesn't work, that there is always an exception to any rule in biology. And don't think that there is an exception to that rule, but maybe I will be surprised. So every concept you find in biology, you will find an exception to it. And so everything is a revolution, everything is a change, everything. But those things teach us about biology, about choices, about why we have one system that works and other options that are available and not necessarily took or not necessarily used. And so I think that the way biologists move science, and I don't think we do just normal science, but we move science by incorporating exception into our rules and sort of accepting those alternative pathways and learning from them new biology, and as I try to show along the way, making those exceptions into tools. So cDNA, CRISPRs, and so on are all ideas that then turn into tools that then were used to study or learn biology and change biology in the way we do biology. And I think I was much over than 40 minutes, so that allows you to get to dinner much faster. And I just need to thank you and funding and my lab members and so on. I did not talk much about my science, but I had a lot of fun making this. I hope you had fun.
We have time for questions, please. Yes.
Yeah, not a question, but it was fascinating, and I think there is something about formulating a thing as a dogma, because even if it turns out not to be true, it encourages a lot of research where people are guided by the wish to find the exceptions you're talking about. Whereas if there was no principle formulated, we would be lost. There were too many things to do. But once someone formalizes
I
the
agree,
principle, then it's
you
very
can actually
useful.
see it in various fields. For example, in the field of cancer, there was a point where there was a review that was trying to understand the underlying mechanism of cancer, sort of view the various ones, and this was used as a theme that, based on that, you can then explore cancer. There was a mimic of this, trying to look at the principle of aging. That one failed completely, or did not fail completely, but just made a mess, and then we're still in a mess. We actually don't have a principle organizing rules for that one, and so we're probably missing something. I think that what was really unique about the way Frick did, or the central dogma, is the ability to create a framework that is so amazing based on so little information. And as you said, this information, trying to challenge this dogma, even if we didn't understand it the way it was he meant it, but still trying to challenge this dogma has actually created a lot of exciting science. And thinking about what you said before about AI, I think that one of the greatest aspects of using an AI is recognizing patterns and in those identifying things that are in a mess of a lot of data. But I think what scientists do that is very different from AI is finding information where no information is there. It's basically creating a picture where all you have is missing part in a puzzle. And you can't deduce what the picture is because you have never seen that picture. It's not like it's a picture of a dog and then I found the tail and I found this and now I can build this picture. I think that we have a picture, we have no idea what that picture is, and we have to guess. And yes, organizing information and pattern can help you. I love information and pattern, but I think that those scientists that do something really unique is sort of like in vivo, it says קפיצת הדרך, you make this amazing jump that it doesn't really clear how you did that, so yeah.
Thanks.
Yes, okay.
Yeah. My microphone doesn't work, so I try to be less loud. Look, Anat, I really liked your lecture. It was great. I know you didn't want to give a lecture, but, but in the end you did, and I'm very happy. And it makes one thing. And one thing is, you said, many people said, a revolution happens when they contradict the central dogma. But then you made it clear that there was no central dogma, at least in the intention of Crick.
Yes.
That it was simply a misunderstanding, and this was his usual showing off or something like that. There are many other things. But it was just a bold hypothesis, and yeah, it led to what you said. And this shows, second, that also what Alan said, that normal science that people are doing now, at least in molecular biology, is quite revolutionary, can be quite revolutionary, and has been quite revolutionary. And you say that Crick in himself was revolutionary when he brought forward the central dogma, because there were so many things not yet known.
Yeah.
But he anticipated it, and it was really a creative kind of creation.
Yeah.
And it was not just following what others already did, and it's not just pressing things into the conceptual tools of professional education, but it was a free thinking and imaginative kind of research. And the other thing I liked was, you said in biology there is no rule without exception. I am also saying that all the time, and that is seems to be such a big difference to physics,
Yeah.
at least to what we heard today about physics. And so one really should be aware of that.
I don't think you can try to find the unifying principle of biology. There is no such thing.
We're not yet that
Principle of
into
biology.
physics.
Yes, please.
Okay, sorry, I have almost no voice, but I think a summarizing principle, I loved your talk, I loved most of them, that I could understand was a difference between deductive reasoning, which is to follow a principle all the way down, that is my sin, that's all I can do, and inductive reasoning. If you read the Jacobin-Monod paper, it is amazing what they concluded about regulation on the basis of no data.
Yeah.
And the same is true for the DNA and RNA, the protein, which still stands, but I never teach it as Crick's dogma.
Yeah, no, I think it's amazing, and when me trying to think what is revolutionary in my mind, is exactly the place where you come up with an hypothesis. Obviously, when it turns out to be true, but in retrospect, where you breach information that is not available, but you create a framework that now can help you plan experiments and get that information.
Thank you, Anna.
Thank you.
So our last speaker